So thank you very much for giving me the opportunity to present some preliminary or some unpublished data that's been coming out of my lab. So just a little bit about my background. I'm a biochemist that's kind of come into evolutionary biology and a little bit into ecology, so please be gentle with some of your questions. But I've kind of really found the field of evolutionary biology so interesting. It's like, okay, let's try and move more into that, and certainly some of the earlier talks about making sure that you do lots and lots of reading is something that I definitely agree with, especially if you're trying to transition from one field to another. So if anyone wants to tweet about me, hopefully nicely, then there's my Twitter handle. And certainly seeing all the tweeting that's been going on about this conference has actually been quite encouraging. At one point it was trending second on the list, just behind Earth Day, and then it fell behind Newt Gingrich. So I have no idea what he's doing at the moment, but obviously he's popular in the Twitter sphere. So I'm going to start off with my acknowledgements, just usually because I run out of time and I forget to do these. And these are the people that have really done most of the work. And I now feel like a faculty member because I can say other people have done the work that I'm presenting. So first of all, most of this work has been done by a very talented undergraduate student, Jeff Wintersinger, who's now at graduate school in the University of Toronto. Former postdoc of mine, Or Gilabert, and PhD student Dave Curran have contributed some of the work that I'm talking about. And also my sponsors. So Jeff received an Alberta Innovates Health Solutions studentship to do this work, recently being funded with an NCERT grant, and also we're part of this NCERT Create training program in host-parasite interactions. And because this is being streamed live, and also being recorded, I should also acknowledge my parents. So hi Mum, hi Dad, thanks. You can now turn off and not see any of the rest of this. So why should we sequence genomes? So we've had talks, and Yannicka was talking about how we can sequence small parts of the genome to try and understand selection pressures, to try and understand population genetics or population genomics. But that's not really good enough for what I do, and potentially isn't good enough for what many people want to do. So my reasons are that I want to investigate evolution at a whole gene level. So whether this is amino acid changes towards a new function or copy number variation. I want to look at functional networks. So here we have metabolism, and that was mentioned earlier. If we want to understand some of the biochemical processes, we really need to get a handle on all the genes that are there. And the best way of doing this is by sequencing the whole genome. And also, as Jason's talk's mentioning, kind of long-range genetic interactions and coevolution. So again, having the whole genome is going to allow us to answer these questions. So it wouldn't be right for me as a genomicist to give a talk without having these graphs on here. So this is the cost of a megabase of DNA sequencing, and this just shows how much it's dropped since 2001, where a megabase was approximately about $7,000 and is now at around 10 cents. And then the cost per genome, so this is based on sequencing the human genome, but we've gone from $100 million down to, that's going to be about $8,000 in 2014. So those numbers are for the human genome, but because the technology is being driven by our desire to sequence the human genome for $1,000, that's meant a lot of us that are working on more interesting organisms than humans can actually sequence the genomes that we care about. So this is numbers from the NCBI genome page from November 2014. But there's about 1,800 genomes for eukaryotic species. So probably many of your favorite organisms are in here. Certainly the ones I work on, so nematode roundworms and flatworms, we have 44 and 13. Actually, by the end of this year, there's going to be nearly 200 roundworms and probably around 50 flatworm genomes that are out there. So the cost is coming down. We're getting a lot more genomes. Lots and lots of fungi. These things are just easy to pump out. So sequencing is no longer the hard part. So what's driving this, this reduction in cost? And it is purely technology. So here, it would be remiss of me to give a talk on main campus at the University of Calgary without pulling up a stickleback. So here's a sequencing machine that was used in 2002. Typically, sequencing centers would have huge hangars of thousands of these sequencers. So if we only had one of the sequencers, it would take about 12 years of this thing running 24/7 for us to sequence the stickleback genome. We move forward to 2015, and here's one machine. It's the Illumina NextSeq. We have one currently in the basement in the Faculty of Veterinary Medicine, and there's more coming. But it takes about 24 hours of continuous sequencing to generate the same amount of data. So sequencing is now relatively easy, and some of my colleagues say sequencing is essentially free. It's not free, but it's a lot more accessible than it used to be. So again, this is related to the human genome, but I think it's pertinent for people working on other organisms. But it was this musing that was in genome medicine, which is we're aiming for the $1,000 genome. So for the stickleback, it might be the $10,000 genome. But actually, it's the analysis that's the bottleneck. So if we want to understand what's going on with all these A's, G's, C's, T's, and quite frequently an awful lot of N's, then we have to spend the money to hire the people to do all the hard work that actually means that we can make sense of it. So once you've had the outlay, so one of these is about $250,000, you can sequence your stickleback, your worm genome for about $10,000. But as I said, it's the analysis, it's the assembly of the genome, it's identifying all the genes, so this is called annotating the genome, but then it's annotating the genes themselves, so assigning function, and then most of you get to the interesting part, which is what's my biological question, how can I actually do something with all these data? So I let other people worry about this, but this is really where the work in my group happens, which is we assemble genomes, or we take other people's genome assemblies, check if they're correct or if they make sense according to us, and then identify genes and assign function. Because typically, if you're sequencing a bird genome, you don't have $100,000 to do the analysis. You get the Genome Sequencing Center to hit go on a genome assembler, spits out the genome assembly. They run a couple of other algorithms identifying genes, assign functions to those, create some pie charts, and off it goes to Nature Genetics and gets published. So our big concern, and I'm jealous because I don't have those Nature Genetics papers, our concern is what do we do once it's being published in Nature or once it's being published in Cell? How can we make sense of it? So just a quick recap on how we do assembly. So you have your in vivo genome, so this is the real genome. You cut it up randomly, you randomly sequence those regions, and then you run a computer program that takes these short reads. Up now, they're about 400 base pairs in length, and you guys may as well consider it a black box. It creates these contigs, and then another black box will take these contigs and build them into scaffolds that you hope approach chromosomes. But in reality, they're only ever a fraction of the size of a chromosome. So how do we measure whether the genome assembly is any good? So many of you will see on genome papers, they record an N50, and as far as, when you hear presentations, people are concerned, a high N50 is good. So what is the N50? The N50 is 50% of the genome on scaffolds that's as large, 50% of the genome is on scaffolds that links to the N50. So in this example, we have a genome that's 1,000 base pairs in length, and then we have all of these scaffolds, and then if we order them from largest to smallest, we then start adding up, and then when we hit 50% of the genome size, so 500, then we know that this scaffold is the N50. So 50% of the genome is on scaffolds of 30 base pairs or greater. So people say, oh, I really want a big N50. It's actually easy to get a large N50. You just tell your program to make my genome as contiguous as possible, and you get a big N50. So an N50 is just really a measure of how contiguous your genome is. So how lenient have you been pulling these contigs together to give these big scaffolds? So it's not really a measure of quality, because some of those contigs could be misassembled, but the N50 doesn't help you with that. So one of the problems is that you don't have a comparison. So you've sequenced your favorite organism, and then you think, well, all I've got is the big N50. I have no idea whether these things are ordered correctly. Well, fortunately, due to various politics, the organism I work on, which is Haemonchus contortus, there were 2 genome projects running simultaneously that published back-to-back in genome biology. And because of politics, we didn't talk about the other one, and they didn't talk about ours. So Haemonchus contortus, Yannick has introduced it really well, but it's a parasitic nematode of sheep. It's the barber pole worm, because it looks like the barber's pole outside the barber shop. Here it is inside a sheep abomasum, and just because in the earlier talk there was a book about laissez fever, well, there's also a book about parasites that you can read your 3-year-old. So we have there's 2 different strains, so there's 2 projects. The first one I'll call the ISE, and the second one's McMaster, and these were the stats, and these were back-to-back papers in genome biology. So the genome size differs by 50 megabases. The N50, so here it's 83 kilobases and 56. The number of scaffolds, 26,000 versus 14,000. The number of genes varies by about 2,000. So these were the number of genes that were predicted. And there's the references. So, and just to disclose, I was part of this genome project. So we decided that we wanted to compare them to say, well, what's going on? One, can we find anything interesting about the biology? Because these are two separate strains, so can we do any population genomics? But equally, why do these two have 2,000 different genes? Why are their genome sizes so different? Which one's better? And we were all hoping for this one, but we were open-minded enough to think, well, if this one's better, then we'll go with that. Or maybe they're both just crap. Sorry, Mum, I shouldn't swear. So this was Jeff's project, 1 species, 2 genome projects, a detailed comparison of 2 Haemonchus contortus genomes. So the initial goal when Jeff came into the lab was, can we detect real inter-isolate variation? However, in reality, it became a case of just how good are these genomes. So the first thing that you typically do with a genome is that you run a Cegma analysis. So Cegma, very briefly, there are 250 or 248 genes that the authors of this paper have determined are conserved across every eukaryote and exist in single copies. So they should exist once and only once in every eukaryotic genome. They had 6 species that included plants, fungi, larger animals, so mammals as well as invertebrates. So, and a complete match of a gene is where you have 70% of that gene present in your genome. So if we consider the complete matches, in ISE, we were missing 32 of these segs, so these ultra-conserved genes, and McMaster were missing 75 of them. So, and the overlap between this was only 25. So we wondered, okay, what's going on? Why are, say, McMaster, for example, missing 75? So we had a look at these 50. We took the seed protein from Seg, so this would have been the protein that was either in C. elegans or in Arabidopsis, and then we blasted against the McMaster genome, and then we used a tool that Jeff wrote called Kablamo, so this is a shameless plug. I recommend everyone use it if you don't know how to visualize a blast search. This is a really cool JavaScript that you can use on the lab website, where up here you have the query sequence. So this is the seed sequence, and then we can visualize where it hits on the subject sequence. So in this example, you can see that this part of the gene, so the N-terminus and then the C-terminus, both match onto a region of the McMaster scaffold. But you're missing this whole middle piece. So this middle piece of this gene is not present in the McMaster genome. And then here's another seed sequence from KEGG where, so here's the whole protein sequence. And as you can see, oh no, sorry, this is the this is the middle piece of this sequence, sorry. And what you can see is it actually appears on a different scaffold and it's completely rearranged. So you have a region here that matches here, and then a region here that matches kind of, it's flipped and on the other side. So this is the reason that the Cegma program couldn't find this gene, is because actually it's been completely misassembled. So if you were doing any analysis and this was a gene that you cared about, then when you went to look at it, you're like, oh, this gene doesn't exist, therefore this worm doesn't have this gene, and then you would start inferring some sort of biology. So the other thing we were able to do, and I said that we didn't have reference, so we didn't know what truth was, but actually we do, because some of Libby's work in the front sequenced a back, so sequenced a large region of the Haemonchus genome, so 410 kilobases, and we were able to take that sequence and then see where it mapped onto our 2 assemblies. So as we can see in ISE, it actually maps across 3 different regions of the assembly, so the in vivo interpretation, and then in McMaster what we see is actually that the front part of the back matches, the middle part of the back matches, and the end part of the back matches, but actually there's these 2 big regions of about 10 kilobases each that we know exist in the Haemonchus genome, but don't exist in this assembly. So that was looking at Cegma, so that was like, okay, they miss either 75 or 32 of these ultra-conserved genes, but how are they if we actually compare the 2 gene annotations together? So we took the proteins from ISE and the proteins from McMaster, and we wanted to see the orthologous relationship. So where do we see a gene in ISE and we see a partner gene in McMaster? Our hypothesis was 90-something% of the gene should be in both of the species. So in the fungi Aspergillus, we see that between strains, 99% of the genes are actually conserved across all the strains. So we think, okay, that's a rapidly evolving organism. Let's hopefully, Haemonchus is slower evolving, so hopefully we should see a score up in the high 90s. So this is actually what we see. So this is where, here's the percentage of ISE proteins that have a ortholog in McMaster. It's only 66%. And then if we look at McMaster and say how many have an ortholog in ISE, it's 45%. These numbers seemed really low. Perhaps it's just a worm thing. Well, if you take two different species, so we have the complete genome for C. elegans and Cystotaxis C. briggsii that are separated by about 20 million years, if we look at orthology there, we see 72% and 68%. So these are two different species. So if we're comparing two isolates of the same species, we'd expect this number to be much greater. And then we also looked at orthology between ISE and C. elegans, and also McMaster versus C. elegans, and we see that ISE, about 60% of its genes have a one-to-one ortholog in C. elegans, or has an ortholog in C. elegans, where McMaster, it's less than 40%. So we were thinking, great, ISE is better, we can just get rid of the other genome and no one has to worry about it, we win. So Jeff then went and did one more piece of analysis that was great and annoying in the same kind of breath. So he looked at these orthologous groups, and typically you expect a one-to-one mapping. This gene has a direct ortholog, sorry, this gene in this genome has a direct ortholog to this gene in this genome. And we see that for 62% of the, for 62% of the groups. But sometimes you will, and some of these could be true, you get a duplication in one species, sorry, in one isolate, and these would be the one to multiple copies or even the multiple to multiple copies. But what's striking is that if we look at groups that have more than one copy in ISE but only one copy in McMaster, that was a quarter of the data set. So this made us think, well, maybe that's because the genome assembly hasn't correctly put together divergent loci that do belong, divergent sequence that actually represents the same loci. What it's done is it's taken these two regions that should be the same gene and said, oh, these two are quite different, I'm going to assemble them into different places so that it looks like there's two genes when there should only be one. And we're still trying to tease out how many cases that's actually true for. So ISE probably is better, but it's certainly not without the flaws, and we've done lots of other work involving gene copy number variation. So ISE wins on most of our metrics, but certainly McMaster does hold its own for some of the other metrics we've been looking at. So we were trying to work out what's the solution, and really the solution is, well, we pour more money into the problem, we do a little bit more sequencing, but actually we devote people to try and get this assembly right. So people started looking at the assembly, they were looking at genes that didn't make sense, and they were saying, okay, these two bits of these two scaffolds aren't correct, let's break them up and reassemble them. So a lot of manual annotation went into this. And the result is an assembly size of 330 megabases. Its N50 is now 5 megabases, so previously it was 83 kilobases, now it's 5,000 kilobases. It's on a lot fewer scaffolds, and we're currently doing the gene annotation. So we have a lot more confidence in this assembly. So I've kind of been talking about worms, parasitic worms, because that's what I work on, but why should anyone care? You know, why should Sean care about his sticklebacks, for example, and the assembly of those? Because surely it's amazing. So I just randomly picked some organisms out of Ensemble and ran Cegma across those. So here we have the three Haemonchus assemblies. So this is the percentage of complete hits we get in Cegma. So again, you need to be 70% of the gene. So we've got 70%, 85%. The new assembly has 94% of these segs, which is great. Partial means that it hits with, it means that 30% of the gene is there. People tend to report partial. I think that's nonsense because only 30% of your gene is there. That's not really useful for anyone that wants to do any sort of functional annotation, but don't let that stop you from reporting it. And this column here is the potential heterozygosity in the assembly. So this is where the assembly method has incorrectly taken two pieces from the same gene and separated them so it looks like you've got two genes. This is probably an overestimate, but it's the best one that we've come up with so far in Cegma. So we had 29%, ISE had 40%, which is huge, and probably explains why a quarter of the orthologous groups are this multiple ISE to one McMaster. But the new version gets this down to 18%, which is actually really manageable and quite nice and allows us to go and individually identify these problems. So if we look at stickleback, 88% complete. So this is the vampire bat. Published genome, 55% complete. Duck, 48, didn't stop it getting into Nature Biotech, I think it was. Sorry if you're involved in the duck genome project and said that it's going to help us with bird flu. I'm sure it will. But you really need to have a better assembly. And then plants don't escape. So eucalyptus and tomato, typically, because of the amount of money that goes into these projects, they do better. And I wouldn't pay any attention to the heterozygosity because of all the big genome duplications that, that go on. But as you can see, these numbers, some of them are okay. So 94% I would say is okay. Anything less, then you're missing core genes that are supposed to be in there. So what happens if you're working on fun genes that are present only in your organism or in your group of, or in your species of interest, and actually undergo copy number variation? So a lot of the genes that people care about, there's 20 copies in your gene or 100 copies in your genome. What about those? So if we're only finding 55% of the ultra-conserved genes, then we're going to be finding many fewer of those that are less ultra-conserved. So I just wanted to give a couple of examples of why people should care and why we care immensely on the quality of the genomes. So these are a couple of analyses that people in my lab have done. So this is Ord's work, and this is looking at conservation of signaling pathways across the whole phylum of Nematoda. So here are the species, and then the columns are the genes. White means that gene wasn't present. Green means that we could use simple BLAST to find that gene. This orangey yellow color means that we had to do something like a little bit of more than blasting. But purple means that we could not find the gene in the assembly using any of the standard methods unless we actually went in there to find it. So a lot of these have been individually cloned, but were not in the assembly. So we know those genes exist, but if you use the annotation that the genome projects give you, you would say that all of these purple genes were missing in these species. So about 10%, 9% of the genes, or of the genes that we know exist, were missing from the assembly. Another example, so this is again a large gene family, the cytochrome P450. So we know that there are about 80 copies in C. elegans, free-living nematode. And if we take the annotation from Haemonchus, we found 2 SIPs. So we didn't think that that was right. So Dave Curran built a hidden Markov model gene finder that's semi-automated, where we take a profile of all of the C. elegans SIPs, model those, and then go searching into the raw genome. And we found 30 SIPs that weren't previously annotated, but we've now annotated as gene models. So, and this actually has a huge consequence to our understanding of the biochemistry of this organism, because if you think you've only got 2 SIPs, you're like, well, how does it survive? And then suddenly you come up with 30 SIPs, you're like, oh, now I understand why it can survive in these diverse environments, from, you know, sheep poo in the grass to inside the abomasum of a sheep. So the summary is that, and I've been a little bit of a grumpy young man or old man about genomes. I just want to make people aware that there's an awful lot of problems there. They're not gospel. You really need to think about your genome of interest. So an assembly really is just an in silico approximation of the in vivo genome. Most of it is mostly correct, so I have a great amount of admiration for the people in genome centers who put these things together. They do it quickly, and most of it is mostly correct. But if you care about your species of interest, you need to spend more time analyzing it, not just taking their gene models and saying, oh, there's 8 copies of this gene in my organism, or it's missing this entire pathway. You need to go and get some bioinformatics skills. That's either your own training or recruit a bioinformatics student, or collaborate with someone. So you really need to ask the questions, are the genes that are missing in the annotation, are they really missing? So this all comes back to taking ownership of your data. It's too easy to go and run some blast searches on your genome of interest and say, this is what's in there. It probably isn't true. So some of the software I've discussed, Figmop, which was that Markov model, Kablamo, which is that really pretty viewer, and also we've just developed a copy number variation tool, all available from the lab webpage. So I've kind of started with my acknowledgements, so I just want to thank everyone for listening, and I'll take any questions. Steve.
Thanks.
That's okay.
Is this working? Oh, there we go.
Yeah.
Great stuff. And it's really good to see someone going in after the fact and asking, okay, how well did we do? So I'm going to ask you a question coming from someone who thinks a lot about population genetics, and all the interesting variations we find among populations, within populations. I've always been kind of worried about, you do a genome and you're done, right? And even if it's a good one, but it's usually, small number of individuals, perhaps one, et cetera. So at what point do you start incorporating the population genetic perspective when you say, okay, we don't have the genome for stickleback, we have the genome of a few stickleback or whatever it happens to be, sheep or gut nematodes. So how much of that is in there right now, I guess?
How much of it is in there right now? It's almost zero. So that's a great question, because it comes back to this point where you really should be building that into your genome project. So if you care about a particular organism, you should be saying, okay, not only do I need $10,000 to sequence the representative, but I need another $10,000 to sequence 100 copies of that representative. Because it's one of these things where your genome project might cost you $250,000, but doing it with population data might only cost you an extra $50,000 because you can map onto that original reference. So actually one thing I did mean to say is talk to your funding agencies. When you write the grants, pay attention to these things. Typically they'll only fund the original genome sequence, and then I've had on grant reviews, well, why are you wanting to do all this other sequencing or all this bioinformatics? We've got the answer. The Broad has sequenced it. Why do we need to spend more time messing around with it? So those things need to be considered right at the beginning, because they're just not in there. Or we have to go after the fact, and it doesn't sound as sexy as sequencing a genome de novo. Resequencing is not as cool, unfortunately. It is, but it's not seen like that.
Thank you.
Nice. Cool. Thank you very much.